As you can see, I've written my first, last name, and the date. My first name is Yash, spelled as Yash. My last name is Monpera, spelled as Monpara. And then I have today's date, which is September 17th. Today, I'll be focusing on Chapter 37, elaborating on the topic protein synthesis and the genetic code. This is one of the topics that many students have been learning in their high school and college level biology courses. The genetic code acts as a blueprint dictating the production of proteins, which in turn carries out numerous cellular functions. Some background information I would like to provide is that adenine, thymine, and cytosine guanine are complementary base pairs found in DNA, where thymine is replaced by uracil in RNA. These nucleotides are organized as three-letter words called codons, which are found on messenger RNA and specify which amino acid needs to be added. Anticodons are located on transfer RNA, which are complementary to codons and regulate protein synthesis. However, genetic mutations can arise because of misinterpretation, which can result in protein defects, further causing various genetic diseases. Genetic information flows from DNA to RNA to protein. DNA is the molecule that stores the genetic guidelines for the functioning of a living organism. Transcription is a process where the section of DNA needed for protein synthesis is copied into mRNA. The mRNA then carries the genetic code from DNA to ribosomes in the cytoplasm, where they read the mRNA sequence and assemble amino acids into a specific protein through a process known as translation. Large mRNA precursors of eukaryotic cells contain coding regions known as exons, which have a job of forming mature mRNA. Even though the central dogma is universal, there are minor differences in the way it occurs. For example, RNA transcripts in eukaryotic cells undergo a series of steps such as RNA splicing, involving the excision of intron sequences, before they can be translated into a protein. In this step, exons are spliced together, resulting in the formation of mature mRNA, which is transported to the cytoplasm for protein translation. Nucleotide sequence of an mRNA molecule contains a series of codons for protein translation. The synthesis of proteins requires around 20 amino acids, which means that there are at least 20 different codons present in the genetic code. I like how Will mentions that each codon must consist of more than one nucleotide to encode all of the 20 amino acids. For example, a single nucleotide codon can code for only 4 amino acids, while a 3 nucleotide codon would go above and beyond and code for 64 amino acids. This would be more than sufficient to code for all 20. It's interesting to note that the initiator codon in mammalian cells, which signals the start of protein translation, is AUG, while UAA, UAG, and UGA serve as terminator codons. The genetic code is degenerate, unambiguous, non-overlapping, without punctuation, and universal. Wheel reports that 3 out of the 64 possible codons do not code for amino acids, and these are termed as nonsense codons. Nonsense codons operate as translation termination signals, specifying where the polymerization of amino acids should end. When we say that the genetic code is degenerate, this means that multiple codons can code for the same amino acid. The third nucleotide in a codon is the least important, and it accounts for most of the degeneracy of a code. For example, 6 distinct codons, UCU, UCC, UCA, UCG, AGU, and AGC, all specify a single amino acid called serine. However, other amino acids such as tryptophan and methionine have a single codon. Something important to keep in mind when studying this course is the difference between ambiguity and degeneracy. When a genetic code is unambiguous, this means that only one amino acid can be indicated from each codon, tRNA-mRNA binding generally tends to be dependent on base pairing rules. Genetic code reading during protein synthesis doesn't involve any overlap of codons, and therefore it's known to be non-overlapping. The message is interpreted continuously without punctuation until a translation stop codon is reached. At least one species of tRNA exists for each of the 20 amino acids. Since there's no binding between nucleic acids, the recognition is accomplished by a protein capable of identifying a specific tRNA molecule as well as an amino acid. The tRNA amino acid charging occurs in two steps, where it's catalyzed by an enzyme for each of the 20 amino acids. The amino acid remains bound to the specific tRNA in an ester linkage until it's incorporated during polypeptide synthesis. One thing that's very important to remember in this course is that the anticodon is read in the direction of 3' to 5', while the genetic code is read in the direction of 5' to 3', as the mRNA and tRNA molecules run anti-parallel to each other during the binding process. The first two bases of the codon and anticodon pair according to the Watson-Crick rules, while the third base tends to exhibit some flexibility, also known as wobble, where a single tRNA can recognize multiple codons for the same amino acid due to the concept of degeneracy that I covered in the previous section. Single mutations result when changes occur in the nucleotide sequence. Single base changes in the mRNA have one of these effects during protein translation. For example, silent mutations can occur if the changed base is at the third nucleotide of a codon. However, codon translation is not influenced by the change at the third position because of the phenomenon known as wobble. A missense effect occurs when a specific nucleotide change results in a different amino acid being incorporated into a protein, and there are two major types of missense effects that I'll be discussing in this section. A partially acceptable missense leads to a protein molecule with partial but abnormal function, while an unacceptable missense effect results in a fully abnormal protein molecule. Another type of genetic mutation is the appearance of a nonsense codon that would lead to a premature termination of translation. However, this has severe consequences as prematurely terminated protein molecules have a high probability of abnormal function. Frameshift mutations occur as a result of insertions or deletions, leading to the formation of altered mRNA and the deletion of multiple nucleotides either prior to or within the nonsense codon would contribute to the reading negatively. Different insertion and deletion combinations resulting in an abnormal protein, as well as being surrounded by normal amino acid sequences, would lead to a number of human diseases. Prokaryotic and some eukaryotic organisms have been diagnosed with abnormal functioning tRNA molecules due to mutations. It's interesting that some of these abnormal tRNA molecules can decode altered codons, suppressing the effects of frameshift, nonsense, and missense mutations. Like transcription, protein synthesis can be described in three phases: initiation, elongation, and termination. mRNA translation typically begins near the 5' end with the formation of the amino terminus of the protein and concludes with the formation of the carboxyl terminus as soon as the message is decoded from 5' to 3'. The formation of the 5' end of the RNA molecule is vital for the commencement of mRNA translation in prokaryotes. The initiation of eukaryotic protein synthesis requires an mRNA molecule to be selected for ribosome translation, and the process begins once the initiation codon is located on the mRNA. Initiation is divided into three steps, which consist of the formation of the 43S pre-initiation complex, the formation of the 48S initiation complex, and formation of the 80S initiation complex. Prior to initiation, 80S ribosomes are dissociated into 40S and 60S subunits during the termination of translation. It's important to understand that there are two tRNAs for methionine, where one is for the initiator codon and the other is for internal methionines. The GTP-EIF2 tRNA ternary complex binds to the 40S ribosomal subunit to form the 43S pre-initiation pre-complex. EIF2 is one of the two major points for protein synthesis in eukaryotic cells, consisting of alpha, beta, and gamma subunits, and this is typically phosphorylated when the cell is experiencing stress. This would occur under various conditions, such as amino acid or glucose starvation, heat, virus infections, serum deprivation, and etc. PKR is actually a very interesting kinase to explore because it can prevent the formation of 43S pre-initiation complex by significantly decreasing protein synthesis through through the inactivation of EIF2B by EIF2A. The 43S pre-initiation complex associates with the mRNA cap and it translocates 5' to 3', scanning the mRNA for an initiation codon, forming the 48S initiation complex. The binding of the 40S and 60S subunits lead to the formation of the 80S complex. EIF4E controls the initiation of translation by regulating ribosome recruitment to mRNA. For example, components of MAP kinase PI3K mTOR RAS and S6 kinase pathways all play a role in enhancing the rate of initiation, while BP1 inhibits initiation. Multi-step elongation. Elongation consists of several steps, such as the binding of amino acyl, tRNA, peptide bond formation, ribosome translocation on mRNA, and removal of deacylated tRNA. The first step is EF1A, forming a ternary complex with GTP and amino acyl tRNA, allowing it to enter the A site. It's important to note that no energy is required, as the amino acid on amino acyl tRNA is already activated. This is followed by the nucleophilic attack of the alpha amino group on peptidotRNA, resulting in the attachment of the peptide bond to tRNA. The third step is the displacement of the peptidotRNA from the A site to the P site, and the fourth one is the departure of the deacylated tRNA from the ribosome. This translocation step leads to the hydrolysis of GTP to GDP and phosphate, speeding up the process of peptide synthesis. Termination. The termination step is fairly simple, which is the recognition of the stop codon by RF1 in the A site. Polysomes, P-bodies, and the protein synthesis machinery. The binding of more than one ribosome results in the formation of a polysome, where the length of the molecule is determined by the number of ribosomes attached. The ER can appear rough due to the presence of the polyribosomes through electron microscopy. mRNAs that aren't part of translation form ribonucleoproteins, which gather in organelles as P-bodies. Ferritin plays a role in preventing ionized iron from becoming toxic within the cells. Environmental threats inhibit the activation of EIF4F and the formation of the 48S complex. Viruses can replicate through the utilization of host cell processes, And many viruses, such as picornaviruses, gain an advantage where they can have protease attack 4G and remove the amino terminal 4E binding site, preventing host cell protein synthesis. Post-translational post-translational processing affects the activity of many proteins. Many animal viruses like poliovirus, HIV, and hepatitis synthesize multiple proteins from a long mRNA molecule. A post-translational modification typically involves the removal of amino acids from the protein through the utilization of specific aminopeptidases. Many antibiotics work by selectively inhibiting protein synthesis in bacteria. Something cool to retain from this section is that many antibiotics only interact with the RNAs and proteins of prokaryotic ribosomes, as they don't have any interactions with the eukaryotic ribosomes. For example, tetracycline and chloramphenicol are two important antibiotics that affect prokaryotes, as tetracycline prevents the binding of amino acid tRNAs in bacterial ribosomes, and chloramphenicol inhibits peptide bond formation. Other antibiotics like pyrimycin inhibit protein synthesis in all ribosomes, while cyclohexamide only affects eukaryotes. Diphtheria toxin is an exotoxin produced by Corynebacterium diphtheriae, responsible for the inactivation of EF2 and the inhibition of protein synthesis in mammals. Now I'll be heading to the Q&A session. What's the correct flow of genetic information? Is it A, DNA to RNA to protein, B, RNA to DNA to protein, C, protein to RNA to DNA, or D, DNA to protein to RNA? The correct answer would be A, DNA to RNA to protein. Which of these antibiotics is responsible for inhibiting protein synthesis only in eukaryotes? Is it A, pyrimycin, B, tetracycline, C, lincomycin, or D, cyclohexamide? The correct answer to this question would be D, cyclohexamide. In which specific nucleotide does the degeneracy of the genetic code occur? Is it A, the second nucleotide, B, the third nucleotide, or C, the first nucleotide? The correct answer would be B, the third nucleotide. Which of these animals are resistant to diphtheria toxin? A, monkeys, B, mice, C, guinea pigs, or D, rabbits? The correct answer would be B, mice. Choose the correct termination codon. Is it A, AUA, B, AUG, C, UAA, or D, UAU? The correct answer would be C, UAA. And then these are just the correct answers. References.